When we think about plumbing, we usually think about the pipes, the solid stuff. But the whole point of a pipe is the empty space inside it. What happens when you need to build plumbing for something really, really small? Like a community of living cells. Building plumbing for a house is one thing. Building plumbing for a single cell, well, that's a whole different level of call the specialist. And the warranty is probably terrible. This is the world of microfluidics and tissue engineering, where scientists are trying to build tiny, lifelike systems on chips. And a paper from a team at Syracuse University presents a masterclass in microscopic plumbing. Their goal is to build artificial capillaries, the smallest blood vessels in our bodies, with unprecedented control. The existing problem is that getting cells to form a perfect, tiny tube is incredibly hard. A common approach is to just mix cells into a gel and hope they self-organize and carve out a path. It's a bit like dumping a bunch of dirt and water in a box and hoping it erodes into a perfect canal system. You get a channel, sort of, but it's messy, the size is unpredictable, and you have very little say in the final architecture. So this team decided to stop asking the cells to be both the construction crew and the residents. Instead, they became the architects first. They used a high-precision 3D printing technique that uses light to create a solid, sacrificial mold inside a block of cell-friendly hydrogel. This mold is the exact shape of the channel they want to create, a perfect, branching network. Then, in a move that's both elegant and simple, they just wash the mold away. What's left behind is a beautiful, empty network of tunnels, precisely defined down to the micrometer. With the plumbing pre-built, they then introduce endothelial cells, the cells that line our actual blood vessels. The cells flow into the network and colonize the inner walls, creating a seamless, living lining. The results are impressive. They report creating controlled channels as small as 35 micrometers in diameter. That's less than half the width of a human hair. They can design straight shots, zigzags, or complex branching networks that mimic real biology. So why does this matter? Because at the cellular scale, the environment is everything. The curvature of a wall, the diameter of a tube, the shear force of the fluid flowing past. All of it drastically changes how a cell behaves. By engineering the emptiness with this level of precision, researchers can finally stop guessing. They can build controlled systems to study how blood vessels work, how drugs get delivered to tissues, or how vascular diseases develop, one well-defined variable at a time. When the group project is to build a bridge, and your job is to design the empty space under it. So, if you could design a custom, microscopic environment for any type of cell, what would you build, and what mystery would you try to solve with it? Let us know in the comments.